Friends, welcome to Biology Made Easy. Today we will discuss in this video the gene interaction, that is epistasis. In Mendel's laws we know that a single gene has two alleles. One is dominant, the other is recessive. And both this dominant and recessive gene, they are present in the same locus. But the dominant one suppresses the expression of recessive one. But when there will be two dominant genes, either in the same chromosome or in different chromosomes, and one dominant gene will suppress the expression of the other dominant gene, that is known as epistasis. Let us explain it. Suppose this is one chromosome and this is another homologous chromosome. Now suppose here the locus, at this locus capital A is there and here small a is there. Both of them are present in the same locus and A is dominant over small a. So this is known as dominance. But suppose let us draw another case. Suppose here in one locus gene A is there and in another locus gene B is there. So A has one expression, B gene has another expression. But suppose this gene A will suppress the expression of B. This is known as epistasis. This is epistasis. It may be present in the same gene, same chromosome, or even it may be present in different chromosomes. So if it is present in different chromosomes, suppose at this locus A is there and somewhere here B is there. But still A will suppress the expression of B. So this is also a case of epistasis, this is also a case of epistasis. That means when one dominant gene present in one locus is suppressing the expression of another gene present in another locus of the same chromosome or a different chromosome, this is known as epistasis. And in epistasis, epi means above and stasis means standing. This is epistasis. Now this epistasis is again of two types. It may be dominant epistasis or it may be recessive epistasis. When the dominant gene will suppress the expression of other gene, that is dominant epistasis. But when the recessive, double recessive genes will suppress the expression of other dominant genes, that is known as the recessive epistasis. And this epistasis first was studied by Beteson. And let us discuss one by one. Let us first discuss the dominant epistasis. And in dominant epistasis, we will take a very common example, that is cucurbita pepo. Cucurbita pepo, which is also commonly known as summer squash. So in this cucurbita pepo, usually the gene capital W is responsible for white color. Capital Y is responsible for yellow color. And when both will be present in their recessive forms, this will be give rise to green color. Now this W is epistatic, while the Y is hypostatic. That means when it is present in WWYY, this will suppress the expression of this. When it is present in heterozygous sperm, then also it will be dominant over it. That means epistatic. But only when it is present in this form or one may be there, then it will give rise to only yellow color. Because dominant form is absent. So if we will make a cross between two white colored summer squashes, what is happening you see? Now let us make a cross between one white with one yellow. white with yellow. So this will be WW small y. This will be small WW capital Y. And naturally the F1 will be capital W small w, capital Y small y. So this is the F1 hybrid. Now what will be the color of this one? Because when both W and Y are present, it is epistatic. That means this also will give rise to white color. White color. Now when two such white colors will be intercrossed, what will happen? Capital W small w, capital Y small y, and another the same one will be, they will be intercrossed. Then in F2 generation what will get? In F2 generation, this will be capital W, capital Y, capital W, small y, small w, capital Y, small w, small y. Similarly, this side also this same way we can write. Now what will happen, you see. In how many places both capital W and capital Y are present? Both the dominants are usually present at 9 places. Because here W and capital W, capital Y there, this means all will be of white color. Here capital W, capital Y are there, that means all will be also white color. Then here capital W, here capital Y, so this will also be white color. Here capital W, here capital Y, this will also be white color. That means at these 9 places, both the dominants are present and all are white color. But you see, here what is there? Here capital W, small y there. So only when capital W is there, this is also white color. Then again you see at this place, here capital W is there, capital W is there, small w, small y. So this will also be white in color. And again this one, capital W, small y. So this will be white in color. That means say at 12 places it is white in color. Where at 9 places both the dominants are there, and at 3 places it is only W is there. So when only W is there, naturally it is white. But when both the dominants are present, capital W is suppressing the expression of Y. So then naturally they will be white. Now see, what about these places? At these places, at the last one, it will be small w, small y. So this is green in color. But what about to these 3? Now these 3, this will be small w, capital Y. This will be small w, capital Y, small y. This will be small w, capital Y, small y. Now you see, here capital Y is present, but W is present only in recessive form. That means say at these 3 places, it will express its own color, which is yellow color. That means now the ratio is 12/16 white, then 3/16 yellow, and 1/16 green. That means it is giving the ratio of 12:3:1, and this 12 is the result of 9+3. Because Mendel's dihybrid ratio is 9:3:3:1. Here this 9 and 3 they have been merged together, because when both the dominants are present, one is epistatic over the other one. So this is known as epistatic gene expression. Now let us discuss the hypostatic one. So in case of hypostatic one, what happens? When the recessive genes are present in homologous form, they suppress the expression of even the dominant genes. So let us take one example, that is the recessive epistasis. And in case of recessive epistasis, usually we get the ratio 9:3:4. Now, one of the very common example of this recessive hypostasis is the lab, or you can call it a hyacinth bean. Hyacinth bean, commonly it is known as same. In this case, there are different fruit colors, and when capital K is present, it is responsible for khaki color. You know khaki color. Then when capital L is present, it changes the effect of K, which is responsible for khaki color, of K to chocolate color. That means when both capital K and capital L will be present, they will also give rise to chocolate color. And when this K is present in lowercase, then it will give rise to buff color. Buff color. Now, if a cross will be made between khaki with a buff color, so this will be capital K, small l, and this will be small k, capital L. That means the F1 hybrid will be capital K, small k, capital L, small l. This is the F1 hybrid. And when both capital A, K and L are there, this will be chocolate color. Now, if this F1 hybrid will be intercrust, in the F2 generation we will get parent square the 16 plants. So, how they are behaving, let us see. That means now the different gametes, this will be capital K, capital L, capital K, small k, capital L, small k, small l. Similarly, this side capital K, capital L, capital K, small k, capital L, small k, small l. So in this case, what is happening, you see, at how many places both the dominants are present. Here both the dominants are present means all this will be both the dominants. Here both are present means all these will be having both dominants. Now here capital K is there, here capital L is there. So this will also be both dominants are there. Then another is this place. Here capital K, here capital L. So this place. That means 1, 2, 3, 4, 5, 6, 7, 8, 9. At these 9 places, 9 by 16 will get chocolate color. Chocolate color. Now, what is happening in other cases? At this place, what is happening, you see? It is capital K, small l. Here it is capital K, small l. And at this point, capital K is there. Capital K, small l there. That means in these 3 places, capital K is there. And when capital K is there, this is khaki color. This is khaki color, this is also khaki color. Now you see at the remaining places. In the remaining places, you see at this position, small k, capital L. Here small k, capital L, small l. And here small k, capital L, small l. And here all will be in the lower form. That means here you see, here both these are present in double recessive form, but L is dominant. Here the same case, one dominant L is there, and here also one dominant L is there. But here this K which is present in recessive form, that is suppressing the expression of L. That is why it is recessive epistasis. So here it will give rise to buff color. So here this is buff color, this is also buff color, this is also buff color, and here it is also naturally buff color. So buff color means it is slightly yellowish gray color. But here you see how the ratio is coming. 9 by 16 chocolate and 3 by 16 it is giving rise to khaki color, and 4 by 16 it is giving rise to buff color. That means it is coming in the ratio of 9:3:4. This is one example. We can discuss also a second example, that is the coat color of mice. So that your understanding will be still better. So let us give the second example, that is coat color of mice. Example 2, coat color of mice. So in this case, the dominant gene C is responsible for black color. If capital A is there, it suppresses the C gene, suppress the C gene and changes to Agouti color. And when both capital A and C are there, naturally it will produce Agouti color. And when the C is present, it is present in dominant form black, but when it is present in recessive form, it gives rise to albino. But when it is present in double recessive form, it suppresses the expression of other genes. That is important. That is why it is known as recessive epistasis. Now, if you will make a cross between two agoutis, so we will cross between two agoutis. This is agouti, another agouti. So it is capital A, small a, capital C, small c. This also will be capital A, small a, capital C, small c. So the F1 hybrid will also be the same one. And this will also be agouti. This F1 hybrid. Now, if the F1 hybrid will be intercrossed in the F2 generation, we will get in the Punnett square we will get 16 plants. Now, how they will behave, you see. Now, here the genes will be here capital A, capital C, small a, capital C, capital A, small c, small a, small c. This side also we can take capital A, capital C, small a, capital C, capital A, small c, and small a, small c. So just like the previous one you see, here capital A and C both are present. That means all these will be agouti color. Here capital C, A and C are there. This means all this will also be the same. Now, here capital C is there, here capital A is there. Both the dominant are present, so that will be agouti color. Then here capital C is there, here capital A is there. It is also the same one. That means at these 9 places, both capital A and capital C are there. So at these 9 places, it will give rise to agouti color. Now you see what is happening in this case. In this case, it will be small a, capital C, capital C. Here it is small a, capital C, small c. And in this case also, small a, capital C, small c. That means here capital C is present alone, but A is present in the recessive form. So when capital C is present, this is black color. So this is black, this is black, this is also black. That means out of 16, 3 by 16 are black. Now you see the remaining cases. In the remaining cases, what happens? Here you see capital A, but C is present in recessive, both. In this case, it is capital A, small a, and C is also again recessive form. In this case also capital A, small a and C also in recessive form. And naturally in this case it is the same thing. It is the all are present in a lower form. That means in these four cases what happens? Here you see this C gene which is responsible for black color when it is present in dominant form. But when it is present in recessive form, it is suppressing the expression of the other dominant gene, that is A. That is why these will all be albino. Albino, all these will be albino. That means it will give rise to 4/16 albino color. That means again it is giving a ratio of 9:3:4. So this is the recessive epistasis. So now I hope you have a very clear understanding what is epistasis, what is dominant epistasis, what is recessive epistasis. Before concluding, I am again repeating that in this same locus, when the dominant one gene or one allele suppresses the other allele present in same locus, that is the relation between dominant and recessive. But when one dominant gene suppresses the expression of another dominant gene present in a different locus of the same chromosome or different chromosome, that is known as epistasis. This is very clear. So I hope you have a very clear understanding. Thank you very much. God bless you. Have a good day.